Hello, Phoenix Biology students. Today we are going to be talking about translation. So I'm going to do a really quick review of some of the stuff that we've already learned and then tackle today's lesson as well. So yesterday we learned about the process of how DNA makes RNA. Now, if you think about it, DNA is like the giant cookbook that your grandma used to have, that red and white checkered thing that had everything in it, from recipes for how to make a cookie, homemade bread, homemade laundry soap, barbecue sauce, how to do the perfect pot roast in the oven. It has a recipe for everything. That's like DNA. DNA is your recipe book for everything your body could potentially need to do. But RNA is a recipe card. It's one single thing that we're trying to make. We're not trying to make everything in the cookbook, so we're only going to make a simplified copy of the instructions for one gene, one protein, one recipe out of that entire recipe book. So when we do that, we're going to simplify things. We're going to use abbreviations. We're going to use the cheapest, fastest way to scribble it down. And that's how DNA operates when it changes into RNA or makes an RNA copy as well. We use that letter U, uracil, instead, and that's because it's cheaper. It's easier to make, it's quicker to use, okay? So when we do make RNA, it can tangle itself up into three different shapes, and I've got the three different shapes here on screen for you. The most common one is mRNA, and the M stands for messenger, and this is simply the instructions. What does DNA say to do? And we're going to take those instructions and we're going to bring them out of the nucleus to go do other things. The tRNA stands for transfer RNA, and this is a type of RNA that is good at transporting things. It's good at carrying things. And the thing that it carries is called an amino acid, and it kind of looks like an upside down cactus or a twisted up triangle, and it always carries this perfect little round blob of amino acids. And as we know, those are the building blocks of proteins. The third type of RNA that we can tangle up and make is called rRNA, and that's what makes up a ribosome to help build those proteins and put them in order. So now that you know the three types of RNA, we're going to move on and talk about, okay, how do we go from RNA, one of these three types, and actually make a protein for our body? So we're going to talk about it in terms of that central dogma that we've talked about before. The first step is that DNA makes more copies of DNA. That's replication. That happens in the nucleus. Then DNA makes a simpler copy of its instructions called RNA. That also happens in the nucleus. And then step 3, going from RNA to proteins, that happens outside of the nucleus at the ribosomes. And if you remember from way, way back, ribosomes can either be found freely floating around in the cell or attached to the ER. So that third step is called translation, and that's what we're going to focus on today. So that step involves two key components. First, we have our mRNA, the messenger RNA. It's just a really simple chain of instructions, and we're going to read that a section at a time. So 3 letters at a time. And each of those little chunks of mRNA, 3 letters at a time, is called a codon. Be careful how you pronounce that. You could get in trouble. I won't tell that story here. But codon. So codon is 3 letters at a time. So when we look at this chain of mRNA, I can't really highlight on here, but you would be looking at 3 of these little letters at one time. So up here, 3 letters at a time, AUG, GCA, and then you'd look at the next section of 3, and each chunk is called a codon. tRNA doesn't have codons, it has what's called an anticodon. So if you think about matter, antimatter, bacteria, antibacterial, they're opposites, right? So an anticodon is the exact matching opposite 3-letter section. And it's always going to pair up AUCG. AUCG. Those are the 4 letters that are present in RNA. So it's going to match that mRNA codon, and it's going to do that to make sure that it's delivering the correct amino acid. We have to do that double check, that proofreading, to make sure that we're not bringing the wrong ingredients for our protein. Okay, I think I got this. So I'm just going to shrink it way down. Sorry that the picture will be small. So on this animation, what you're going to see is the entire process of the RNA being made into protein. So here we've got the nucleus. It looks like a weirdo planet in the background. And coming out of the nucleus is the mRNA, the messenger RNA. We've just made that simple copy of our recipe, and now it's floating out in the cell until it meets a ribosome. The ribosome is going to grab on at the start of the instructions, and it's going to be pulled along, kind of like an old school cassette tape or a VHS tape is pulled through the machine. The mRNA message is pulled through the machine. And as it goes, we're going to try and match each of those codons to the anticodon on these guys. These are the tRNAs, and each one has the little red dot on it, and that little red dot is an amino acid. There are 20 different varieties of amino acid, and we can assemble all the proteins we ever need in our body by rearranging the order of those amino acids. So as you can see, the tRNA is always the same. It just carries a different amino acid into the ribosome. So if we look, you can see that it's coming through kind of choppy because we're reading 3 letters at a time. We bring in a tRNA that is an exact match to these 3 letters down here, the codon anticodon match. Make sure that we're bringing in the right thing. If it is the right thing, it can drop off its amino acid. If it's wrong, it gets kicked back out again. So as you can see, as they drop off their amino acids in the correct order because they've checked the codon-anticodon match, it starts to build a chain of amino acids. And that chain of amino acids comes out of the ribosome, and that's our raw protein. Whether it's a pigment for your skin, a protein like a digestive enzyme, or hundreds of other examples, we just assemble it one little piece at a time. And we're going to read that entire recipe, the entire mRNA message. And when we've finished, that protein that's finished is going to be released from the ribosome, and it's going to be sent out into the cell to be folded up and processed in just the right way. And the protein that we've made real time in that little example was hemoglobin, which is a protein on your blood that helps you carry oxygen. So now let's take some notes on it. First step was that the ribosome had to find something called a start codon. Codon is 3 letters, and the start codon is the 3-letter sequence that tells us this is where to start cooking. This is the first step of the recipe right here. And in every living thing, in all of biology, it is the exact same code, AUG. Now, that would be amazing if every single computer program, software program, everything had the exact same code for this is where you click to do this one thing. But every program is different. So it's kind of a miracle, actually, that everything in biology starts with the exact same 3-letter code, AUG. It's going to be kind of important. The next phase, as we saw, is that these tRNAs have to start dropping off and bringing in the correct amino acid pieces. So the mRNA is pulled through the ribosome. Remember, mRNA is yellow, ribosome is this blue blob back here. And we're going to focus on each of those codons, 3 letters at a time. AUG was the first one that told us where to start. CCC was the second one. When we got that, the matching RNA, tRNA, drops off an amino acid. And then we do it again. So for GAC, here comes our big tRNA, drops off the right amino acid. And then we move over. And we keep going and going and going until we build that chain of amino acids. The last phase is to stop and process that RNA. We reach something else called a stop codon. The stop codon could be a couple different ways of telling this ribosome, nope, you're done, and then the protein is released. The protein goes through some folding and some packaging, a little bit of just fancying it up, making sure that it's ready, and then we can use that protein in the cell. So as practice, I've got a short little video here for you. And I will link that in this if I can. I gotta figure out how to do that. So I'll link that separately just in case the sound doesn't come through. But you're going to have an amino acid codon chart given to you for all of the homework and all of the things like that. But the basics are you start at this center section and you work your way out. So let's watch this in process. So here's your amino acid codon wheel, and our first codon is CCA. So if we follow that down from the center, CCA leads to proline. So I would write in P for proline. Whoa, I'm getting dizzy. The next one is GGG. So I start at G, go to the next G, and then the third G, and that leads me to glycine, which I abbreviate G. Go to the next one, which is CUC. I start here in the center with C, and then go to U, and then the third C, and that leads me to a different amino acid, leucine. So if you think about this kind of like Lego building block instructions, you've got the 3 letters CCC. GGG, whatever they are, and they tell you what piece you need to add next. The last one here is UUG, so I'm going to find UUG, and that leads me to another leucine. So there's lots of different instructions, and sometimes they repeat themselves and create the same product. Okay? So our final answer here, proline, glycine, leucine, and leucine were the 4 amino acids that these codons told us to create. So all 3 steps in order. If you are doing DNA replication, you're making more DNA. So that's going to partner up like this. DNA across from other DNA, it's going to partner up A's and T's, C's and G's. The other option, if you're making RNA in the process of transcription, when you're going from DNA double helix to an RNA simplified recipe, you're going to partner up slightly differently. Across from A is U, T is A, C is G, G is C. So this is the weird one. We have to replace the T's with the letter U. And when you're going from RNA to protein, you're going to read 3 letters at a time, A, C, C. You're going to use that little wheel chart thing to help you find out what is our amino acid that we've built, and then we add those together to make a chain of protein. So I hope this is helpful to you. I hope it helps you with the homework, and I hope it helps you on your test. Make sure that you're taking notes today on your guided note sheet to help you remember and recall all of these steps in order. I wish you guys all the best. Good luck with everything today. I look forward to connecting with you tomorrow.